Our next presentation titled Untargeted Metabolomics Identified Key Metabolic Pathway Altered by Thymoquinone in Leukemic Cancer Cells, presented by Dr. Asma Alghamdi. Dr. Asma Alghamdi received her Master of Science from Biochemistry at King Abdulaziz University. She is interested in medical genetics, epigenetics, and hematology as a field to pursue her career. Her research interests lie primarily in the area of is the plant medicine and therapeutic product and developing the drug delivery and cure genetic and epigenetic disease. Dr. Asma Alghamdi, you can start your presentation. Thank you.
You are welcome. You can hear me?
Yes, you can start, please.
Okay. Assalamu alaikum wa barakatuh, and good evening to everyone. I would like first of all to thank the organizer of this conference for accepting my start. Let me now introduce myself. I am Asma Alghamdi. As you can see on the screen, my topic today is untargeted metabolic identify key metabolic pathway altered by thymoquinone and leukemic cancer cells. Next slide, please. Okay, in my presentation, I will focus on 5 main sections: introduction, aim of the study, experimental design, result, conclusion. Next slide, please. Because the failure of the types of treatment used in some cases of leukemia, modern research has tended to treat cancer by use of alternative treatment. One of these alternatives is thymoquinone. Thymoquinone, an artery-organic anti-cancer compound extracted from Nigella sativa oil. Thymoquinone showed anti-proliferative, pro-apoptotic, and anti-metastatic effects of different cancer cells. Next, please. Metabolomics is the study of cellular metabolism at the global level. Metabolomic study can lead to enhanced understanding of disease mechanism and biomarkers, enhanced understanding of mechanism for drugs, increased ability to predict individual variation in drug response phenotype. Next. The study was aimed to explore the global metabolic impact of thymoquinone on cancer cells, hematological cancer cells, using untargeted metabolomics. Next. In experimental, we used 2 cell lines, JORKIT and HL60 cells, and treated with various doses of thymoquinone. Metabolomics was done by using LC-MS bioinformatic analysis. Next. The metabolome profiling that changed by thymoquinone treatment, measurement by extracted the cell using a combination of acetone methanol solvent. Then injected the sample into LC-MS. The total metabolite identification is 335 feature metabolite in HL60 JORKIT cells. 302 features show p-value less than 0.01. Figure A show the BCA performed on all the samples, which closely cluster to one another. Figure B is heat map indicates thymoquinone have over effect in the metabolic variation on both cell lines. We found a dramatic increase in thiamine glycol, a metabolite recognized to induce DNA damage. Also, we observed a strong decrease in cellular guanine levels, is really suggestion alteration in nucleotide metabolism. Next. In cellular pathway, the apoptosis is a major contribution in maintaining tissue homeostasis. Thymoquinone changes the cell metabolic rate and induces apoptosis. In HL60, phenylalanine metabolism show higher impact by thymoquinone treatment. It activates OFROC and it induces apoptosis. Also, linoleic acid metabolism change cell proliferation and induce apoptosis. In JORKIT cells, arachidonic acid metabolism, higher variation and changed by thymoquinone. Another higher impact by thymoquinone treatment is tyrosine metabolism that interlink with other metabolic pathway like glutamine and tyrosine kinase, which is downstream activation of apoptosis. Next. We identify some pathway and component that affected by thymoquinone treatment. One of these pathways is TCA metabolism and amino acid metabolism. Thymoquinone changes the metabolism compound in TCA cycle. The K metabolite in TCA cycle is glutamine, is converted to alpha-ketoglutarate. Glutamine in HL60 cell shows significantly reduced by thymoquinone, but in JORKIT significant change not observed. Alpha-ketoglutarate is a acceleration of cell proliferation by thymoquinone on both cell lines. Diethyl fumarate observed to reduce its circulation by thymoquinone on both cell lines. Next. In methylation, cysteine and methionine metabolism play a major role in DNA methylation. Thymoquinone treatment has high impact on metabolites responsible for DNA methylation and epigenetic modification. Metabolite homocysteine and L-methionine observed reduced accumulation by thymoquinone treatment in both cell lines. There was heterogeneity between HL60 and Jurkat cells, but thymoquinone interfered epigenetic modification in both cell lines. Next. The conclusion of our study, we first time showed the metabolic landscape of cancer cells during the thymoquinone treatment. Thymoquinone globally alter metabolome of cancer cells. Thymoquinone treatment alter primary nucleotide metabolism and promote metabolites involved in genomic damage and ultimately cell death. Finally, our research is dependent on hadith of Prophet Muhammad, peace be upon him. He said, use the black seed, which is a healing for all disease except Assam, and the Assam is a death. Next. I want to say thank you to all co-authors Dr. Hani Chaudhry, Dr. Muhammad Imran Khan, Dr. Mazen Zamzami, Prof. Muhammad Gari, Prof. Abdurrahman Malki, and Muhammad Rajid. Next. If you have any question you would like to ask, you can send to my email. And for more information about paper, you can check my page in ResearchGate. Next. Thank you for listening to me.